Hi, my name is Shay Chemistry with the Dana Foundation, Career Network and Society. Today I'm really excited to be joined by Dr. Jeyifous. Would you like to introduce yourself?
Yes, thank you for having me. I'm Dr. Oak Jeyifous. I'm an assistant professor at DePaul University in the Department of Neuroscience, and this is my third year. So I'm a relatively new hire, but yeah, that's my primary, appointment. And then I also am a neuroscience. I'm a bioarts, or I have a bioarts practice that I've been sort of working on and focused on for the last few years as well. So I'll be talking a bit about how I've merged those two interests and working on those two kind of in a formal way.
Thank you for being here today and being willing to take some time to talk to us.
Okay.
My first question for you is that, are you able to tell us a little bit about your career path and how that led to doing work in the intersection between neuroscience and art?
Yes, absolutely. So I think um, both my interest in science and art started at a fairly young age. Like in terms of the arts, my mom was a photographer, and she had a very strong interest in visual arts and in architectural design. And so my brother and I, both from like a very early age, were subjected to, going to galleries, and we had like a lot of art in the house, and was something that was stressed, and then we spent time actually sort of talking about and um and then we had friends of our family who were practicing artists, or like professional artists, and so it was just always around and kind of part of how I grew up. And then in terms of the sciences, I think that I also had a very keen and a keen interest in kind of the natural world, or like the biology of the natural world. So like in animals and in landscapes, flora, fauna, from like a very early age. And I think that this led to kind of me being interested in certain biological questions and processes. And then when I got to college and kind of could take formal, classes in neuroscience, that's what really sort of sparked my interest in kind of the brain and how the brain functions, and trying to figure out how we how we sort of perceive our world, and build a representation of things that are happening in our world. Like, what are the mechanisms that like allow that, right? So uh these two, I think, for a long time, um kind of developed, or I saw them as being separate entities, right? So I have an interest in art, and that's here, and I do things, towards that interest, right? And I was big into photography, and sort of had this a burgeoning kind of photographic practice, but I saw that as being separate from my science training. So I'm, learning how to do science and how to think critically about science, but I never sort of thought about how those two formally um, might, intersect, right? And I think at some point when I was in graduate school and a bit after grad school is when I realized that some of that my aesthetic eye, or the principles that I use to sort of interrogate space, or look at a macro piece, a portrait or a landscape study, or a photo of like a building, that the way in which I um, looked at that piece and looked at the space and thought about, structure and composition, was very, very similar to the eye that I use to evaluate like a microscopic field. So when I'm looking at neurons in a dish, I'm similarly looking for structural relationships, nuance change, and in the structure and shape and size of neurons in a dish, and that I was utilizing kind of some of the same principles, the same physical concepts, right? And that's when I was like, oh, like maybe there's a way in which I can more formally kind of interrogate both of these at the same time. And that's when this sort of came about, is that I became interested in utilizing my research tools. So tools I use in the lab, microscopes, reagents, as like vehicles for, um, you know, for making these kind of novel visual layers and tapestries, and coming up with ways in which I could study or make comments on non-scientific principles using scientific tools. And that's kind of like the first, the first kind of major project I did involved doing just that. And so then I was like, oh, like I can actually, in the context of doing my research, kind of utilize these tools to create some novel visual piece that is not tied to my research, in like a direct way, but that allows me to investigate certain themes, that may be science related or maybe are not science related. And then I think beyond that, it became really clear that this could be a really effective tool for talking about science to audiences that maybe would not normally sort of engage with, someone who was doing science research on learning and memory or drugs and abuse. So it's a means of doing science outreach and having conversations with people who may not think about the research or ever sort of ever encounter that research. I can now discuss scientific principles and concepts in the context of a visual piece that actually may resonate with them, right? So I saw the potential for this as like a means of science outreach and in engaging with the public more broadly around scientific themes, but doing so in a way that was more digestible, maybe, or that may reach more people, right? And so that's been the kind of transition from kind of like, this having an early interest in both of these things, in both of these pursuits separately, realizing that for me, they actually are very much one and the same in certain ways. And then I utilize many of the same cognitive tools and, creative tools to do the work. So why not merge these two in a cool and novel way that like allows me to do more than I would with each of those singly. Right? Yeah.
It is a really fascinating journey, and I think a lot of students are going to be able to relate to wanting to combine their multidisciplinary interests and figuring out how they can channel both audiences and combine both of those interests.
Yep.
I think my next question then is to ask, how do you, how would you explain the day-to-day life juggling, working with the art practices, but also neuroscience? What does that look like to you?
Yeah, that's a great question. I think that that's something that is still very much kind of a work in progress, right? Um, I'm a relatively new faculty member here, and so what that means, is that I had to sort of get used to teaching formally again and in course design and, trying to do a lot of work around becoming a better teacher and, becoming a more, effective teacher, and that takes work. So I think that I came here and I had sort of certain plans or a certain timeline with respect to my bio art stuff. And that, of course, was, has like gone by the wayside. Like it's, tough the first year or two to have all the time to do everything. I'm a, parent of twins, and so I have to spend time doing that, obviously. I'm starting my lab up and then trying to teach. And so all that said, I'm in my third year now, and so I've gotten better at time management and at trying to carve out, in like um, a very sort of purposeful way, and very, um, in a how would you say, like in a a deliberate way, time to actively sort of work on these things. So for me, it meant trying to incorporate aspects of this bioarts practice into my formal workflow. So as I'm doing, so, during the week I teach on Tuesday, Thursdays most of the time. And so I have certain time on Monday, Wednesday, Friday to work on research or to train students in the lab or to do sample preparation. And then I tend to do like my work on microscopes on Friday, Saturday, or Friday, Saturday, Sunday. So I have certain days during the week where I'm utilizing certain tools. And so I quickly learned that if I can incorporate aspects of my bioarts workflow into my regular sort of sample workflow or research workflow, that's a way to make progress on these projects. And so that's what I do is that, if I have Monday, Wednesday, Friday, I have some sample prep time. I will try to work into that same workflow, um, processing those samples, staining cells, or, whatever it might be. And then at the end of my regular, imaging session, I might have time to scan some of these samples that are meant for like my bioarts project. But I have to be very, um, uh, I had to incorporate it in like a formal way into my regular work schedule on like a weekly basis. Otherwise, it's very easy to have things kind of linger for months on end, right? Or just to focus on these things during the summer, which is what I had done the last 2 years. But then now, like I said, I've worked on a way to incorporate this into my month to month work, week to week work. and that's made it a lot easier to make progress on some of these projects, right? So um, I'd say that you have to really um, force yourself to incorporate it, into your weekly schedule, or it's easy to sort of have it slide away, right? So that's how I do it now. It's just basically um, I've had to sacrifice some of the more the working on these things sort of as they pop up versus scheduling time to be creative. So it's like a bit of a different way to do this arts practice in that like I'll have, an hour here, a couple hours here, and I have to switch into that mode
Mm.
and maximize that time. And I've gotten better at doing that, kind of, picking back up where I left off the week before or a couple weeks before. And if I have this 1.5 hour slot, I need to come in here and really sort of focus right away and switch into like the visual mode. Um, but that's been effective the last half year or so. So
Yeah, that's great advice. I think kind of what you said is about being deliberate and intentional with your time and making sure that, it stays on your mind and fitting it in where you can
absolutely,
substantially.
absolutely.
Do you have any other further advice that you would like to share with future students who are interested in doing neuro art work in the future?
Yeah. So I think, as I've sort of well, as I've talked about, that seeing these, two things, so science and art or neuroscience and art, seeing them as an endeavor that can be merged was like huge, right? So when I finally came to that realization that some of the same sort of muscles I use to think about a scientific problem or to come up with a methodology for asking and answering a question in the context of my research, that skill is not separate from the skill that I would use to sort of develop a bioarts project or to think about how to push or how to investigate a certain theme in like a visual way. It's the same skill set, right? So once you come to that realization, I think that really helps you come up with new ways to like expand the work and to push it further. It certainly did for me. I you tend to see less, or tend to place less um less sort of what would be the term, like you're not as um rigid in your understanding of kind of what is possible utilizing these like tools. So like it became a lot easier to come up with new ways to um process and create these visual layers and tapestries once I was taking into account, all the tools I have available to myself, right? In the context of my research lab, et cetera. And I think also, um, so there's that, and then also the scheduling aspect. So just a time aspect where um, being you have to ensure that you have um, time that is dedicated to doing this work and to think specifically about sort of how to expand this, practice. And that has to be done in like, a very deliberate way, right? And most importantly, probably at this stage, is making yourself aware of or informing yourself as to sort of funding opportunities or training resources that might be available to students that have this interest. So when I first started, there were not a lot of sort of funding opportunities or residencies and things like that, things that sort of help to really drive the artistic practice. That's changed drastically over the last sort of decade, right? So um, there are residencies that now specifically ask for or are looking for people that have ideas that lie at the intersection of science and art, which means that there are now more funds available to do this work, right? So finding out what those are, what the and really formalizing this, right? There are also now many more graduate school programs and training programs that are focused on this, new kind of, intersection of science and art. So there are many opportunities at different levels of like analysis that involve working at the intersection of neuroscience and art, and that's starting to pop up in graduate training programs. So if you're an undergrad student and know you have this interest, find out where those are. Are there people and or programs that might speak to your specific interests? Um, really any way to formalize that work, to, give you deadlines, to give you exposure to and or a greater number of resources that will, in like a very practical way, allow you to do that work, one. And also just formalize it, right? The more you do it, the better you become at it. And you will also then meet people who are also similarly thinking about those kinds of questions. So I've been greatly helped by conversations with working artists and also other bio artists, and that's been very, very helpful and not only shaping my work, but in giving me new projects and or new things to think about. So meeting people, talking about your work, and exploring these themes in like a very formal way, I think is critical. Making the time to do it and trying to maximize some of these new training programs and funding opportunities that are out there for people that have this interest. Right.
That was amazing. Thank you so much for sharing and giving that valuable insight that I think a lot of students are going to be able to learn something from and, get inspired. I think that's all we have for today. So just thank you for being here.
Thank you so much for the opportunity. Had a lot of fun talking about this.
Okay.